Hello, my name is Joshua Mugabi. I welcome you to Biology King. And in this session, we are going to talk about Introduction to Cell. Outline the three components of the cell theory. 1. All living things are composed of cells. 2. The cell is the smallest unit of life. And 3. Cells rise from pre-existing cells only. Describe the future of the striated muscle fiber that challenge the concept of the cell theory. Muscle fibers form fused muscle cells and hence are long and have multiple nuclei. The cell is the smallest unit of life. Describe the features of the giant algae that challenge the concept of cell theory. Certain species of unicellular algae may grow to a very large size. That is, for example, greater than 7 centimeters in length. Challenges the idea that cells are microscopic and larger organisms are composed of many cells. Describe the features of the septate fungal hyphae that challenge the concept of cell theory. Filamentous hyphae that are not partitioned, are septate, will have a continuous cytoplasm. Challenges the idea that living structures are always composed of discrete cells. List the functions of life. Metabolism. Living things undertake essential chemical reactions. Reproduction. Living things produce offspring through sexual and asexual. Sensitivity. Living things are responsive to stimuli. Homeostasis. Living things maintain a stable internal environment. Excretion. Living things require the removal of waste products. Nutrition. Living things exchange material with the environment. Growth. Living things can move and change shape or size. Compare the metabolic functions of Paramecium and Chlorella. Of the diagram of the Paramecium and that one of the Chlorella. The mode of nutrition in Paramecium is heterotrophic, whereas the mode of nutrition in Chlorella is autotrophic. Paramecium engulf food, that is via phagocytosis, and absorb essential gases via diffusion. Chlorella contains chlorophyll for photosynthesis. Paramecium Solids are removed via an ionophore, while liquids are pumped out via virtues. Chlorella, all essential materials are both absorbed and eliminated via passive diffusion. Explain the importance of surface area to volume ratio as a factor limiting cell size. The rate of metabolism of a cell is a function of its volume, mass over volume. The rate of material exchange between a cell and its environment is a function of the surface area. As cells grow, its volume increases faster than its surface area, meaning metabolic rate eventually exceeds the rate of exchange. Hence, cell size are limited in order to maintain viable surface area to volume ratio. List 3 adaptations of cells that may function to maximize the surface area to volume ratio. 1. Cells may adapt to an elongated or flattened shape to increase the surface area to volume ratio. For example, the bacillus. 2. Cells may possess cellular extensions. We can see that one in root hairs or folds to plasma membrane. Those ones can be seen in microvilli. And third, cells may reduce effective cell volume by including large central virtues. Example, we look at that one in plant cells. Suggest with a reason the location of the following cell within the human body. The location is the intestine or the digestive tract. Reason is it has microvilli to increase the surface area, which allows for more material absorption. Compare the advantages of using light microscope and electron microscope. The light microscope can view living specimens, whereas the electron microscope has a magnification. Those are advantages. Light microscope can view natural colors, whereas an electron microscope has got a higher resolution. Compare the disadvantages of using a light microscope and electron microscope. The light microscope has got a lower magnification and it has a lower resolution. For the electron microscope, can only view dead specimens and views in monochromes or use false colors. Identify the difference between a transmission electron micrograph and a scanning electron micrograph. The term generates cross-sections through a specimen, that is the internal image, whereas the same, it generates a surface rendering of specimen, that is the external image. Calculate the actual size of the following image, whose width is 11 centimeters. And we can see on the image, it has a magnification of 86. The actual size is given by image divided by magnification. And the image size is 11 centimeters. The magnification is 86. So the actual size will be 11 divided by 86, which will give us 0.128 centimeters. So the actual size will be 1.28 millimeters. Calculate the linear magnification and actual size of the following image. Scale width is 2.2 centimeters, that is the image. The parasite width is 7.2 centimeter, that is the image. Therefore, magnification is given by the image divided by the actual size. So magnification will be 22,000 divided by 50, which is going to give us 440. So the actual is always given by the image divided by magnification. So the actual size will be 7.2 divided by 440, which gives us 0.0163 centimeters. So the actual size will be 163 micrometers. Outline with an example the organization of cells in a multicellular organism. Now, cells of the same type may group together to form tissues. For example, neurons, they form the nerve tissues. Different tissues interact to form functional organs. We have the heart, which is composed of the nerve and cardiac muscle tissues. The organs combine to form organ systems. For example, the heart and the blood vessels, they form the cardiovascular system. Define emergent properties in relation to multicellular organisms. An emergent property is a function present in a multicellular organism that is not present in any of its individual component cells. Emergent properties arise from the interaction of individual cells to produce new functions in multicellular aggregates. An example is increased antibiotic resistance seen in biofilms. Explain how cells differentiate through selective gene expression. Now, all cells of an organism contain an identical genome. What we are saying, the entire set of genetic instructions for that organism. Now, differentiation involves the expression of some genes and not others in the cell's genome. The activation of certain genes by chemical signals will cause a cell to develop differently from other cells. Differentiation through selective gene expression causes different cell types to become more specialized and distinct as they mature. Describe how genes may be packaged within eukaryotic cells. Now, the DNA in eukaryotes is packaged with proteins. Those are the ones that we call the histones within the nucleus to form chromatin. The active genes are usually packaged in an expanded or accessible form called euchromatin. The inactive genes are usually tightly packaged in a more condensed form, which we call heterochromatin. And the packaging of chromatin varies between cells according to which genes are active and inactive. State the two key qualities of stem cells. One is self-renewal. Stem cells are capable of continuous cell division. And second, it is potency. Stem cells are capable of differentiation. They are effectively unspecialized precursors. What are totipotent stem cells? With examples. Now, totipotent can form any cell type and form extra embryonic structures. That is, they can form new organisms. For example, we have the blastocyte. What are pluripotent stem cells? You give also examples. Pluripotent can form any cell type but cannot form extra embryonic structures like the placenta. For example, the embryonic stem cells. What are multipotent stem cells? Also give examples. The multipotent stem cells can differentiate into closely related cell types. For example, the hematopoietic cells in the bone marrow, those ones form a variety of blood cells. What are unipotent stem cells? I want you to give also examples. The unipotent, these ones can divide but not differentiate. For example, muscle cells can self-replicate. outline the therapeutic use of stem cells. In the diagram we are here seeing the structures. We have the bone harvesting the cells, then they differentiate and then can be implanted. 1. Stem cells can be used to replace damaged or diseased cells with healthy ones. Stem cells can be harvested from embryos, umbilical cord, blood, or certain adult tissues. For example, the bone marrows. The biochemical solutions are used to trigger the differentiation of stem cells. Then the cells are surgically implanted into the patient's own tissue, and the host immune system is suppressed to prevent rejection of the cells. So there is harvest, inducing to differentiate, and then implantation. The new cells must be monitored to ensure they do not become cancerous. Those are the new cells. Describe the use of stem cells to treat Stargardt's disease. Stargardt's disease is caused by gene mutation that impairs photoreceptor cells in the retina. Now, this leads to genetically inherited macular degeneration or resulting in blindness. And it may be treated by replacing the damaged retinal cells with a healthy one, those ones which we derive from stem cells. List 3 other therapeutic examples of stem cells. Now, they can be used to replace dead nerve cells in individuals with Parkinson's disease. Parkinson's disease causes the death of dopamine secreting neurons. Secondly, they can replace the bone marrow of individuals with the leukemia, that is the cancer of the blood. And they can be used to replace the pancreatic beta cells in individuals with the type 1 diabetes. Those are the ones that cannot regulate the blood sugar levels. Compare the implications of stem cell therapy using cells derived from different sources. We have cells that can be got from the embryo, the umbilical cord blood, and then adult tissue. So we shall look at their growth potential, tumor risks, harvesting, and disadvantages. Let us start with the growth potential. The embryo, the growth potential is high. The cord blood, it is high. That is the growth potential. And adult tissue, the growth potential is low. Now, when it comes to risks, tumor risks, for the embryo, it is high. The cord blood, it is lower. And when you come to the adult tissues, the tumerics are low. Now, what of harvesting? For the embryo, this can be generated artificially by SCNT. Then from harvesting from the cord blood, this one is easily obtained and can easily be stored or preserved. In adults, it is difficult to obtain and invasive to extract. Now, what are the disadvantages? For the embryo, it requires the destruction of the embryo. Then for the cord blood, cells must be stored from birth, and that one will require a high cost. Then for the adult tissues, they may be limited in scope of application. Outline how stem cells may be artificially derived by SCNT. Now, this involves the creation of embryos via therapeutic cloning. That is, fusing a diploid nucleus with an enucleated egg cell and then stimulating with an electric current to promote division. So, there are excess embryos that are created by this process, which raises the ethical concern about the destruction of the embryos. Outline how stem cells may be artificially derived by nuclear reprogramming. There is induction or induce a change in the gene expression profile of a cell in order to transform it into a different cell type. This involves the use of oncogenic retroviruses, increasing the risk of cancer. Hello, my friends. My name is Joshua. I thank you very much for participating in Biology King. Please, I encourage you to subscribe.